I'm going to turn off my video so improve the bandwidth.
Just getting the input sources working. Yeah. Do it. Yes. No. Yufei, I haven't heard from. Okay. Here's my slides. I'm going to do a screen share. Screen share. Oh boy. On my other desktop. Click on there, and I'm going to do slideshow.
Oh, it's from 9 a.m.?
From 9 a.m. That's right. I got to send an email to my committee. Make sure they show up. Ooh, font is small. Need some kind of props so I can see this. Oh, speaking of the devil. Hello. Welcome. Gary, are you there?
Yeah.
Oh, right. Sorry, Gary, are you there?
Yes.
Okay, sorry. We're having a trouble with the sound. Let me play around with that for a minute. It could be the settings.
I'll be right back.
Okay.
Yeah.
It's not this one here. No, we used this one last time.
Yeah, I think there should be an audio cable that connects from here, right?
I see, but oh, we were in the other room yesterday. I see.
Yeah.
Okay, I think we're okay to just have the area on the small speakers. Okay, so we're gonna just have you outputting on my local speakers, not on the big speakers. Cool, I think we're ready to go. And I've already got a slide done. 65 to go. Great, so welcome. My name is Ben Cipollini. A lot of people on campus know me as the guy who cares about lateralization, which is a good thing because I care a lot about lateralization. And what I noticed when I was putting this presentation together is that actually grad school is like so lateralized. Everything we do seems to involve lateralization. We do a lot of talking, which is lateralized, and writing, that's lateralized. A lot of manual skill, for example, when our softball team won the championship, a lot of manual skill, throwing and hitting, all lateralized. We learned to read a lot of new words, like which is left lateralized. We also learned to recognize a lot of new faces in people at conferences and in the department, right lateralized. Even kind of getting lost in the details of what we do and losing the big picture is lateralized. And yet, in my lab, even in my lab where we study vision, I don't think we really appreciate how fundamental lateralization is to what we do. So if a stimulus is presented, if a subject fixates at a central fixation cross and the stimulus is presented to the left of that fixation cross, the information will go wholesale to the right hemisphere. And vice versa, presented to the right will go to the left hemisphere. But if it's the normal case where you get a stimulus presented centrally, that stimulus is actually cut in half, and half of it goes to each hemisphere. And then it's up to the hemispheres to communicate with each other at some point to figure out what that stimulus is. And I will tell you, and you'll hopefully see in this talk, that that communication is not straightforward. But none of the models we have actually account for the two processing streams of the two hemispheres, and the fact that they do different things. So today's talk is not about lateralization. I think we fundamentally underestimated the role of interhemispheric transfer and how it plays a role in lateralization. So today I'm going to focus on the interaction between lateralization and interhemispheric communication, and I'll address it through two approaches. First, I'll be proposing an integrated hypothesis on visual processing asymmetries and how it involves asymmetries in the brain and interhemispheric communications. And then I'll focus more on interhemispheric communication itself and the idea that brain size affects interhemispheric communication and therefore lateralization. Okay. So I'll start with a brief introduction into visual asymmetries. There are two major theories trying to cover the scope of visual asymmetries, and their overlap is surprisingly sparse. So the first theory here, by Berman and Plaut, is driven largely by cortical representation of visual objects that we're experts in. Today I'll focus on faces and words. Faces and words show lateralized activity in the fusiform gyrus, so the ventral part of the temporal cortex, and with faces showing greater activation in the right hemisphere, words in the left hemisphere. They share homologous regions of the temporal cortex, the corresponding areas. Crucially, lateralization doesn't appear until about 5 years of age, and it seems to be, in the lateralization of words and faces seem to be interdependent. So lateralization in face processing is correlated with reading experience and verbal skills, and Marlene Berman has led the way in kind of investigating the relationships, the push and pull between visual word forms and faces in typically developing children and typically developed adults, and also adults who haven't developed typically. So congenital prosopagnostics who have a deficit in face recognition, or developmental dyslexics who have a problem with phonological processing. The theory, the basic theory on the right here, is that words use both. Not quite yet. Words use and faces both use high acuity central representations. So that's this area down here. They're both sharing the central representation. They need high acuity vision, but they are very different types of representations. And so there's kind of competition between them. And so this competition is represented by these arrows. The idea is that word forms become left lateralized because language resides in the left hemisphere and is more efficient to represent them closer to phonetic language. And then faces become right lateralized because the left hemisphere is kind of occupied by the representations of visual word forms. And so competition occurs specifically when children learn to read. If you go to cultures where they don't learn, where people are illiterate, you won't find this strong right and left lateralization. Okay, another seemingly separate but classic literature shows lateralization of processing these Nabhan stimuli, these local T and global level T, or spatial frequency grading. The high spatial frequency grading with the left hemisphere preference, low spatial frequency grading with the right hemisphere preference. Often these are used in lateralized presentation, and asymmetry seems to occur when stimuli are in low contrast or presented very quickly. So in this model on the right, the encoding of stimuli is actually symmetric between the hemispheres. What's asymmetric down attentional weighting of that encoded information. So information's selected based on the task at hand, and the right hemisphere shows a bias for lower frequency information in that frequency band, and the left hemisphere for higher frequency information in that frequency band. The hypothesis I'll present today attempts to combine the data and ideas from those two hypotheses and to extend it by proposing a specific neuroanatomical mechanism behind it. In this model, the hemispheres encode information differently. This is driven by differences in the representations, the spatial representations of that information, where things like natural images, faces, low frequency gratings, and contours all share some kind of representation, but things like words have a different type of visual representation. So very similar to the Berman and Plout model. The right hemisphere is better at, the hypothesis is that the right hemisphere is better at finding these kind of local spatial correlations that exist in the face and natural image data, and it's driven by a difference in long range, in the spread of long-range lateral connections in cortex, and its developmental trajectory is governed by interhemispheric transfer. So I'll present 3 simulations and try and give some description to help support this hypothesis. So the name of the model I'll be describing is the differential encoding model, and it's a model of long-range lateral connectivity. Now, in my experience, not a lot of people in cognitive science or even computational vision are aware of what long-range lateral connections are or what they do. So I'll just start by describing what they are and what they do. What we're used to seeing is connectivity between regions of cortex. These connections, these white matter connections that sit just below the surface of cortex. And we can discover these through, for example, diffusion tensor imaging and tractography, something there's been an explosion of over the last 10 years. But you can actually inflate the cortex. It's really a very thin sheet, and so you can inflate it like a balloon and flatten it out. And if you do this and then you inject dye into the cortex, into that thin sheet, you'll see labeling that looks patchy like this. And what you find is that there's a dense region of connectivity at the place where you injected the dye, suggesting really strong local circuits, local connectivity. This is a cortical column. You'll also see in select regions of cortex, not all of it, but just very sparsely across cortex, other columns, other patches that are labeled, suggesting that they're interconnected with the area, selectively interconnected with the area that you injected dye in. And so if we take this labeling scheme and put it into a representation of visual cortex where different columns or different neurons have a receptive field, and it maps the entire visual space, you can just see that the red is the injection site and the green represent connected neighbors.
Is that in vitro or in vivo?
This is ex vivo. This is after you die. And
So then this stuff happens. Is the spreading of that dye is.
yeah, I mean when you're injecting dye, usually you've sacrificed an animal. So it's something that was living and sacrificed, and yeah. So this is just been mapped onto a play kind of digital retinotopic visual area. And what long-range lateral connections are thought to do is, in the red position, you think you have some feature, you think you have some orientation, right? But the stimulus is kind of weak. You can use information from your neighbors to say, well, what are your orientations? What do you think is going on? And then collaborate to try and strengthen your belief that you got the right orientation or not. The kind of thing that happens in contour processing. And across all images, you can try and pull out common contours that go through this region of the cortex. And so that's what this really simple neural network on the right is doing. It just does a weighted sum of its green connected neighbors and updates its belief, its activation, that some feature is present or not. And we can find these correlations among a bunch of images by creating what's called an autoencoder neural network, where you have a bunch of these hidden units, one per pixel. So there's 850 pixels, and there will be 850 hidden units, each with this kind of sparse random connectivity. And if you train this neural network to just reproduce the input image, it will learn correlations among its connected features, looking for example, contours that are in common across the image set. And so in this model, we've got two parameters. One is the width of the Gaussian to sample from. Those connections can be very widespread, or they can be very narrow. And the number of connections to, per hidden unit here is essentially 6, 5 neighbors, and yourself. And so the training paradigm is you create the network, again, choosing the spread of connections and the number of connections, and you select an image set. So here I have 4 Navon figures. And then you present one of the images. You get the activity forward. It's noisy. It hasn't learned well. And so you backpropagate that error to adjust the weights so that you can do a better job of reconstructing the image. Next time, that's still not quite good enough. And so we'll just keep training. Now the output image still has error, but we've hit our error criterion, and so we stop training. And then we can just present the image again, and we can analyze the output image itself. We can see, in the error, is there any preference for spatial frequency? Is this network representing some spatial frequencies better than others? And we can also grab these internal hidden units, which represent, features discovered by this connectivity paradigm, and we can use these representations to run through behavioral tasks, and I'll describe that more in a bit. So I use this same paradigm for 3 simulations with slight tweaks. So in the first simulation, I try to account for visual development. So during development, we know 3 things. First, visual acuity and contrast sensitivity both start out poor and improve over time. So visual inputs are relatively blurry and biased for low frequencies. Second, there's quite a bit of literature suggesting that the right hemisphere begins maturing earlier than the left hemisphere, suggesting that the right hemisphere will get more low-frequency inputs than the left hemisphere while it's maturing. And the third is that these patchy connections mature by changing their connection strength, but also by pruning, by removing connections that are irrelevant, that are not used well, and that's depicted on the right-hand diagram. And so overall, the idea is right hemisphere will prune connections under blurrier, lower spatial frequency input than the left hemisphere. And so I took one of our neural networks. Here I've depicted one of the 850 hidden units. And I've got 2 networks with the exact same connectivity. But I train a natural image patches, but I vary between the 2 hemispheres how blurry the images are during development, with the right hemisphere receiving blurrier images than the left hemisphere network. And while training, I'm not only going to strengthen the connections, change the connection weights to be stronger, but I'm going to remove the weakest connections, just like that happens during anatomical development. And the only difference between the networks is that the right hemisphere network is trained on blurrier images than the left hemisphere network. And afterwards, after the pruning, I may have some different distribution of connections between the 2 networks, and that's what I'm going to analyze. So here's
Sorry.
no problem. Do you need a minute, or are you good?
No, I'm good.
Okay. So here's the.
Hang on just a second, sorry. John, we're in the middle of Ben's practice thesis defense. Okay.
She wants to join. Give her
Yeah.
the YouTube feed. Um, so here's just a depiction of the training schedule for what inputs the right hemisphere and left hemisphere are receiving. And you can see that the right hemisphere is receiving blurrier inputs than the left. You can see actually that these two match. And then at the end, in the last stage, the networks will be trained to some error criterion. So they'll still learn equally as well. It's just the right hemisphere essentially starts learning earlier. When I do this, and after training and after pruning, I take all the hidden units and I just enter them at one location, and I get the empirical distribution of connections after pruning. And I get them for the right hemisphere network trained on blurred images, left hemisphere network trained on less blurry images. They're different, and so to visualize the difference, I just subtract. And what you see is that at the center is greater representation of connections for right hemisphere network, and in the periphery is greater representation for left hemisphere networks. It says that shorter connections are encoding low spatial frequency information better. Great. That's simulation 1. In the second simulation, we're going to focus on one of these behavioral paradigms. How does this connectivity pattern affect behavior? And so in the behavior, again, they use lateralized presentation where stimulus is briefly presented to the left or right of the fixation cross, and as I mentioned before, it crosses over to the contralateral hemisphere. And if you present the same stimulus to each hemisphere, to each visual field, and therefore to each hemisphere, if you see differences in reaction times to the task, you can get some indication that there's a processing difference in the hemispheres. And so for the task I'm looking at here, the task is just to say, did you see a target? Doesn't matter if it's big or small, did you see a target? Press the button. If so, the targets are T and H. And so if the target is at the larger global level, like the T here, you'll see faster reaction times for the right hemisphere, for left visual field presentation. If the target's at the local level, the smaller level, you see the opposite pattern. You'll see faster reaction times for presentation to the left hemisphere than the right. And so to model this, we use the same networks. We trained them on these Navon images. There were 16 of them, two targets, 2 distractors. After training the autoencoders, we extracted these hidden unit encodings, and then on a separate kind of classification neural network, we trained it on the behavioral task. Here's the hidden unit representation of the large T. Do you see a T? Yes or no? And then we see which neural network, or we see which neural network learns if there's a difference in their learning based on these different representations. So here again is the human data, and here are the data on the error on the output of the left hemisphere and right hemisphere networks. And you can see we replicate the target level by hemisphere interaction that you see in the human behavioral data. So this suggests that the networks with this connectivity structure are storing that the connectivity structure biases the information that's being stored internally in the network, with a right hemisphere preference for the global level information, left hemisphere preference for the local information. The third simulation is actually taking the same data, so that the same networks, but instead of analyzing those hidden representations, I analyze the output. I need a slide here. The output images. And I do a spatial frequency decomposition of those images, and I just have plot them spatial frequency versus the original image to look for error, right, between the right hemisphere's output images and the original image, left hemisphere's images and the original image, and then I compare the two. So this is basically goodness of fit, with right hemisphere preference, or better representation in the right hemisphere above the 0 line, better representation in the left hemisphere below the 0 line. What you see is for the lower spatial frequencies, you have better representation in the right hemisphere. For the higher frequencies, better representation in the left hemisphere. if I just selectively take these spatial frequencies and I just show those, I filter out all the others, you can see the right hemisphere seems to be focusing on the global level information. And if I focus on just these left hemisphere preferred frequencies and eliminate the others, you can see that the left hemisphere's representation seems to be focusing on the local level stimulus. I can do the same paradigm. I can train these networks on things like faces or natural images, or even natural images that have been put in a log polar representation, trying to simulate the warping of cortex due to foveal overrepresentation. And in all of these cases, you see that the output images, the right hemisphere network represents low frequency information better than the left, and the left hemisphere network represents high frequency information better than the right. And so we have 3 simulations showing an association between right hemisphere short connections, global level of the stimulus, and low spatial frequencies, and the left hemisphere with longer connections, the local level of the Navon figure, and high frequencies, high spatial frequencies. So there's many reasons, many other reasons to think that visual asymmetries are related to long-range lateral connections, and I don't have time to discuss them. So to summarize, what I will show you is that the right hemisphere seems to be specialized for encoding more local spatial relationships than the left hemisphere. This is due to shorter long-range lateral connections in the right hemisphere. I didn't show you, but in retinotopic visual areas later than V2. And the connectivity asymmetry comes from development where visual acuity and contrast sensitivity changes interact with asymmetry and developmental timing, and leads to differential connection pruning. I mentioned that these images have different types of spatial representations, and so I'll show you just one possibility of how that may be. And so what I've done here, and I should have an image for this, what I've done here is I select one pixel from, say, a face, and then I just correlate it with every other pixel in that image over the entire set. And I see how does spatial correlation differ as you get farther and farther from any pixel, across all the pixels, averaged over. And what you see for natural images and faces is really high predictability, high correlation at very close neighbors. That drops off as you get farther from that pixel. You see that for natural images, and you see that for faces. But if you look at words, you see a very different pattern. You see that this dot here is correlation of a pixel with itself, so it's always 1. But its neighbors doesn't correlate very strongly with. And the weak correlation actually spreads out pretty far. And so you can imagine how connections that are very local are picking up relationships, right? Correlations between nearby pixels, and ones that are farther away are not picking up relationships, but weaker ones. Whereas for words, whether you're close or far, you're getting weak correlations anyway, and the structure is anisotropic. It's just in one direction. In the vertical direction, you're getting no correlation whatsoever. And so the shorter connections may be biased for maybe picking up the natural image, the local correlations there, and the wide ones do a poorer job, but both do an equally poor job on the words. And that may be one reason why you get this.
If you do that with Chinese.
Yeah, so this is an interesting question. I haven't done this. I did this last night. But there are definitely differences in processing and asymmetry in different languages. And so that's a great empirical question. OK. So I just told you that thin connection asymmetry comes in early, like by 15 months of age, while this connection pruning is happening. But I also told you that in Berman and Plaut's theory, or actually in their data, asymmetry seems to come in later, starting at 5 years and continuing through 14 years. So how to resolve these two differences? And I suggest that this can be done through the development of interhemispheric transfer. And so the idea is that in later visual areas, interhemispheric transfer is weak until around 5 years of age, continuing through 14. This is a hypothesis, this is a question, so the question is why? Why is interhemispheric transfer weak during development? And I'll just point out here, here's a sagittal view, sagittal view of the corpus callosum. And you can see the corpus callosum contains many different fiber diameters, right? It's a heterogeneous structure. And what I'm going to talk about is how that heterogeneous structure changes over development. So I'm going to take a short interlude to talk about interhemispheric development. And I'm going to start with just a statement of some work I did with Gary that I presented at CogSci last year. What we found is that conduction delay magnitude, despite what is written in the literature, is computationally irrelevant. Okay? Conduction delays seem to matter only because they correlate with things that actually matter. For example, conduction delay correlates with distance, and distance correlates with white matter volume. And white matter volume increases faster than the volume of the brain itself. So you have really strong pressure to minimize your white matter volume, because even as it is, at some point in brain growth, the brain will be 99.99% white matter volume. It's taking over. No other quantity does this. Conduction delay and length also correlates with metabolism. The brain takes up 20% of our energy. There's really high constraints on metabolism. And finally, conduction delay magnitude correlates with variability in conduction delays. And that's what I'm going to focus on here. And I think what I'm going to talk about is not going to be familiar with many people, because I haven't seen anybody in the cognitive literature talking about variability in conduction delays. In all the simulations I've seen, they're assumed to be perfect every time. So let's see. There's evidence in the literature that small unmyelinated axons with a diameter of less than half a micrometer have unreliable delays, up to 20% variability in the timing of that spike. And that delay is dependent on the length of that connection. And what you can see is that during development, the diameter of axons initially starts out small. What's in this red box are all axons that are 0.5 micrometers or smaller in a cat. And you can see that prenatally, the preponderance of fibers are these thin, unmyelinated fibers. That after birth, the distribution starts to widen out, and you have many but fewer of the unmyelinated, thin, unmyelinated, noisy fibers. And then finally, into early adulthood, you get myelination, and also many of the fibers become wide enough to become very reliable. I don't have time to talk about it today, but there's good reasons to think that the degree of this effect is potentially unique to humans due to the fact that we have really big brains, but they're very highly compressed brains in order to give birth without creating new connections after birth. And so the question I asked in this work was, what are the effects of delay-dependent variability? And what we found is that reliable delays don't interrupt long-distance communication. So you've got a left and right model hemispheres here, and these interhemispheric connections have long delays. If those delays are reliable, there's no interruption of interhemispheric communication. If those delays are unreliable, then you get a bias for local intrahemispheric circuits. And this matches what you find in development, an early bias for local connections, local circuits, and then with development, spreading out to wider ones. So this offers a mechanism. I haven't heard any other mechanism for this, and this offers a potential mechanism for explaining that. And that reduction in variability during development leads to greater hemispheric collaboration with age. Oops, sorry. And what I see in the differential encoding model, going back to the developmental one, is that when the hemispheres are independent, and I'm training them on these frequency biased images that eventually become full fidelity, the asymmetries actually are reduced with experience. Even though you're pruning those connections along the way, by the time you've trained the model fully, the asymmetry in frequency representations is almost none. So this suggests that independent hemispheres have less asymmetry. And to be honest, I think this makes a lot of sense. When you're completely independent, you're dependent on yourself. You have to solve every problem yourself. You can't count on anybody else, right? But when you have others around and when you communicate with them, then you can specialize. Then you can, be the programmer and I'll be the farmer and whatever. Someone's gonna, build the highways for us. This kind of thing happens in all sorts of systems, like multicellular organisms. Independent single-cell organisms can survive on their own, but if I take a red blood cell out of my body or a skin cell, it's just going to die. It's depending on others, and it has cellular communication mechanism for working with that specialization. So I think that all makes sense. That independence would lead to less asymmetry, less specialization. Okay, so going back to the visual asymmetry hypothesis, using this information about interhemispheric communication, here's the timeline for the hypothesis. At 3 to 15 months, a connection asymmetry emerges from connection pruning. After 15 months, pruning ends, and further visual experience of relatively independent hemispheres suppresses those asymmetries. From around 5 to 14 years old, these later visual areas in the fusiform area connect with the fibers that they connect with start maturing at that stage. And that maturation enables interhemispheric communication and therefore specialization. And the latent connection bias that wasn't really affected during the earlier time leads to right hemispheres to learn faces better and contours and low frequency information faster than the left because it has that connection bias, and the right hemisphere will learn the words faster. Moving on. 10 minutes. So in the second part, I want to talk about the relationship between brain size and lateralization. So I just argue that the hemispheres are interdependent, but for a lot of people, it's obvious that the hemispheres are independent. For example, before brain surgery, you might inject some chemical into someone's carotid artery on one side, and that will essentially disable or turn off one hemisphere. If they do that, they still act relatively normal. If you inject the left hemisphere, they won't be able to talk, but they never report. They never freak out. They never say, "Oh my God, the world has changed. I'm an alien. Who am I?" Nothing like that. Second, for split brain patients where the corpus callosum is cut, and the two hemispheres in adulthood, and the two hemispheres can't communicate anymore, we'll just ask. And so there's a special with Alan Alda where he asks this callosal patient, JW, "Do you feel any differently now when you think?" And JW says, "No, I just got a backed up brain now is all." So these kind of things suggest that the hemispheres are independent. They seem to be working without each other, without communicating. And so the question in the literature has been, does brain size affect lateralization? It seems like, well, it must. I mean, we have big brains, and we think that makes us human. Our brains are lateralized, and we think that makes us human, because it involves language and handedness and crucial things like that. So there must be a relationship. There are two papers that really hone in on these arguments, not just from intuition, but arguing that actually this is true, that there's less interhemispheric communication due to a reduction in the proportion of interhemispheric fibers, and then reduction in the conduction delay magnitude with brain size. I think looking into the literature, I think there are really good reasons to question these hypotheses, though. First, anatomical and functional asymmetries exist in large brains, but also in small brains. So even the lowly toad has a lateralization in Still don't remember I have to look that up. Second, humans that are born without a corpus callosum. So these, you can think of these as like really reduced number of connections or really, really long connections, right? That take a really long time to communicate. They don't show greater lateralization, as would be predicted by the model. They show pretty typical lateralization. Some typical lateralization, some reduced lateralization. During development, delays decrease, but lateralization in language, and as I talked about, vision and handedness, all increase. And finally, if you look at functional connectivity, so just allowing someone to sit in the scanner and measuring their brain activity and seeing how brain activity at different parts of the brain correlate with each other, you always see the highest correlations between these homotopic areas across the hemispheres as compared to just any other comparisons you can make, suggesting that the hemispheres may be coordinating very closely. Okay. So in my research, I've taken a second look at each of these. I'm just going to summarize the findings from the first. I talked about them briefly before. In this neural network model, which runs with delays, all the delays are short, one time step, except the delays across the hemispheres can be short or long. What they find, or what I find in the model, is that there's a short delay onset of interhemispheric communication with long delays, and nothing more. The two hemispheres still communicate with each other equally. There's just a short onset. And I think that this onset difference is probably irrelevant. First, in the model, the callosal and interhemispheric delays are drastically exaggerated, 10 times longer across the hemispheres than within a hemisphere. Whereas in a real system, I might guess 1 to 3 times more. And this is on the order of like 5 to 20 milliseconds, something on the brain's processing time, not that long. Second, the corpus callosum doesn't contain one speed of connection. It contains many delays that interconnect areas. And it could be that these really short delays are exactly for initiating that communication and overcoming those delays. The model doesn't capture that. I think that's a critical component. And third, the model has no predictability. But if you can predict, have a good guess of what is coming on the order of 5 to 20 milliseconds, so it's a really short time to predict, right? It's not that. It's likely you can predict, at least with some certainty, what's coming in the next 5 to 20 milliseconds. If you can predict at all, you can always overcome delays. And so I say this model shows no evidence of independence with larger brain size. And again, I don't even think independence would lead to lateralization anyway. I want to focus the rest of my time on this second study, which uses allometry to examine the proportion of interhemispheric fibers across primate species. And so I assume most of you aren't familiar with allometry, so I'll give you a very brief primer. The basic idea of allometry is that big brains are not big versions of little brains. Brains change with brain size. The architecture changes to compensate for things like delays, to optimize for metabolism, to make sure that big brains aren't very slow and then therefore can't compete with small brained animals who are much quicker. And so, for example, if the brain was a sphere, you would expect the relationship of the surface area being 2/3 power of the volume, because volume goes as r cubed, surface area is r squared, so the relationship should be 2/3. But in actual brains, you see that the relationship is 8/9. There's more surface area for a particular brain volume. There's more surface area than you would expect for a smooth sphere. And the reason is, as brains get bigger, they go from smooth, in general, they go from smooth to more wrinkled, to more wrinkled, to more wrinkled. And the reason that's done, well, at least one big reason, is what I told you before about the white matter volume. By folding the brain, you can actually reduce the volume of white matter connectivity, right? Because you just bring the cortex closer together. you can also increase the number of neurons that you have in the brain and potentially increase computational power. And so allometry essentially works on scale-free power laws, or power laws. So in Euclidean plots, these would be, there are three types. One is sublinear, so when your exponent is, your power law exponent is less than 1, it means that what's on the x-axis is growing faster than what's on the y-axis. When they're linear, it means they both scale at the same rate with brain size. And when your exponent is greater than 1, it means what's on the y-axis is scaling faster than what's on the x-axis. And you can plot these on log-log axes, and you'll get lines for each. And so your line will have slope less than 1, so the slope is essentially the exponent. So slope 0.5, slope 1, slope 2 here. And you can just use linear regression, basic linear regression, to examine these things. And you can see this, these laws apply to microscopic properties like neuron density, which decreases with brain size. So as brain gets bigger, the neuron density in the gray matter decreases. Or, as I told you before, macroscopic quantities like white matter volume. Yeah. And these are actually, these three in blue are ones that I'm going to use in the analysis I'm going to do. So the original study took MRI, full brain MRI volumes of 11 primate species, and what they essentially wanted to compare is the total number of fibers in the white matter fibers in the brain versus the number of fibers, those white matter fibers that are interhemispheric, that are callosal. What they were able to reconstruct from the MRI volumes are an estimate of the gray matter surface area and the callosal surface area. And so the idea is the surface area, gray matter surface area, is close to the number of neurons, which is close to the number of connections. And the callosal surface area is, represents the number of connections you can fit in that area. And when you compare those two quantities allometrically, so you do a linear regression on the log-log plot, you can see it fits a line pretty well. And if you plot that back out to the Euclidean, you can see this is slightly sublinear. And that says that what's on the x-axis grows faster than what's on the y-axis. Surface area or total connectivity seems to grow faster than callosal or white matter, or callosal surface area or connectivity. I think there's a few reasons to doubt the conclusion from this analysis. First, the missing quantities like the neuron density or the callosal axon density, things you would use to calculate the number of connections, those also change with brain size. And so the relationship, you can't just make that mapping. You actually have to make the estimate of connections. The second is I think the results are confounded with how with a different level, a higher level of structure in the brain, where the cortex is separated into areas, and the number of areas increase with brain size, and the number of these connections increase within a hemisphere. Inter-area connections increase within a hemisphere, but across the hemispheres it doesn't change with brain size. And so the number of fibers in one of these bundles may stay the same within a hemisphere and across hemispheres. It's just that you have more of these bundles with bigger brain size, and so it kind of washes out any individual connection. And I'll try and describe that a little bit more carefully. So in this small brain, you've got 16 areas, 8 in each hemisphere. In the large brain, 32, 16 in each. Each area has 4 inter-area connections, and here it's got 8, so the number is increasing with brain size. But crucially, even though kind of this interhemispheric connection is scaling just as all the rest do, right? It looks like there are proportionally fewer connections because it's more of these different interhemispheric connections proliferating. But this isn't something special to interhemispheric connections. If you take any intrahemispheric area connection, it also seems to scale with to be reduced with brain size. And so I don't think comparing the total number of connections is the right way to look at functional connectivity, because we wouldn't want to say V1 to V2 is less important with brain size. Instead, I think we want to compare the number of fibers within one of these connections to see if one of these connections are selectively reduced compared to each other. Okay, so we need to do two things to accomplish this. First, we have to estimate the total number of fibers and total callosal fibers rather than surface areas. Here's the really simple equations for doing that. You just take the callosal area and multiply the axon density in the callosum, take it the number of connections, and you take the total number of neurons times the number that tend to project into the white matter. Total number of neurons is just the volume, gray matter volume, which we have, and the neuron density, which we have in the literature. So when I pull that data out of the literature, I plug in and I have 3 quantities in, and I'm missing one. Fortunately, in the literature, there are data that are good enough to use for this. I can estimate. And so I pull off these data. You can see it seems to go an exponential law. Here's the allometric fit. You see it's well fit with a power of minus 0.28, suggesting that density of fibers decreases with brain size. In other words, the fibers are getting bigger with brain size. This human data is not part of the regression, only through the macaque side. The human data was taken from the literature and plotted in there, and you can see that the human data fits that regression very well, suggesting that this is a good regression to use for this analysis. And so when I plug that all in, here's the original estimate with a 0.88 power. I get something that's a much more shallow slope that drops off much more quickly. Our estimate is that the total number of callosal axons, increases with 0.63 power of the total number of connections. The total number of connections is scaling way faster than callosal connections. And so the question is, it's not even close. Why? What's going on? So as I said, we have this compound of the number of inter-area connections within the hemisphere increasing at the same time. How much of this difference does that account for? And so what I do is I plot this ratio of interhemispheric inter-area connections versus intrahemispheric inter-area connections. This is going to be 1. This is going to be increasing with brain size. And when I see how does that scale with the total number of callosal connections versus the total number of fibers, or total number of callosal fibers versus the total number of fibers across the whole brain. And if these relationships are the same, then I expect isometry. I expect the power of 1, and they will all the scaling difference here will be explained by this difference here. And that's basically what I get. I get a power of 1.02. So that's as close as isometry as I would ever hope for. Now, because they scale with isometry, I can actually do a little algebra, and I can compute the relative number of connections in a callosal inter-area connection versus an intrahemispheric one. And I find that callosal connections are like 4.3 times larger. They have 4.3 times more fibers than your average intrahemispheric area connection. And this is true even though the number of connections in the fiber connections tends to decrease with distance. So the farther away those areas are, the fewer fibers there are in there. And these connections, these callosal connections, are very distant, but still show this relationship. So I don't think convection delay magnitude shows independence. I don't think the proportion of fibers suggests independence with greater brain size. I don't think independence suggests lateralization with greater brain size. And so I think we've been headed down the wrong direction with this literature. To summarize, in the Rilling and Insel paper, they thought the proportion of interhemispheric fibers decreases slowly with brain size, and this suggests a functional decrease. What I found is that the proportion of interhemispheric fibers drastically decreases with brain size, but there's a compounding factor that explains all of that variance, and that actually there's no functional decrease in callosal communication with brain size, and instead it actually suggests that callosal fiber bundles may be stronger and special compared to intrahemispheric. So to conclude, I suggest that development and interhemispheric communication drive connection differences that lead to visual asymmetries, and there's long-range lateral connections. I offered a mechanism for the developmental bias towards more local circuits, conduction delay variability. I stated, essentially, didn't really show evidence, but stated that conduction delay magnitude likely does not cause independence, and that independence is unlikely to cause lateralization anyway. And finally, I showed that callosal connectivity is robust across brain size, including humans, and there's some evidence that corpus callosum plays a special role. From here, I've got a couple of projects I'm looking at doing. One is to create an integrated model where I have interhemispheric connections with the connection asymmetry of the differential encoding model, and put that through a developmental trajectory and essentially try to model all of visual development in adulthood in a single model. The second is I've been looking at using allometry to estimate how, from animal data, how the human corpus callosum changes over development and into adulthood. We have very little data, but these data will tell us a lot about what we should expect from things like DTI. We don't know what the axon diameter distributions are in young adults or even children, and so we might be able to estimate that from animal data. So I'm looking at that now. Oh, interesting. And that's all. Just want to thank everybody. That's it. Thank you. Okay, any comments? Shorter than yesterday.
Yeah, you've still got about 10 minutes to go to cut it.
Yeah.
So I guess, I mean, you can cut a little bit at the beginning. You know, I think it's cute to talk about your soccer team or whatever it was, but you know, I think you could just say that in a few bullets and get it over with.
A few bullets. Go ahead.
Yeah. I wouldn't say that your lab doesn't understand how important lateralization is. And then I wouldn't say that this isn't about lateralization. So why did you bring up lateralization if your talk isn't about that? So.
Well, it's about the two together, but yeah, I hear you. It wasn't that clear.
And then you said lateralization doesn't appear until 5 years of age. Well, what about language?
Yeah, visual.
Okay, well, so there is lateralization, and that's why a lot of Berman think that words go to the left.
Right.
Put a circle around the little t, and the big T.
Mm-hmm. You're reading notes.
Huh?
You're reading notes. Can you send them to me when you're done? Thank you.
I was surprised that you said that your model combines Ivory and Robertson and Platt and Berman. I guess, I don't know, maybe I lost focus for a little bit, but I'm not sure why. What about Platt and Berman's idea do we add to our theory?
What about Platt and Berman's?
How does Platt and Berman's idea contribute? What component of your theory is the Platt and Berman theory?
Yeah, the competition between the hemispheres, the developmental, all their data on the developmental delay, and the idea that the representations for words and for faces are just different.
Yeah, but where is there competition between the hemispheres in your model?
It's not in the model, it's in the theory.
Okay. Well, I don't like theories very much. Okay. Are you gone? Hello?
Cool.
Hi.
Sorry.
It's okay. Anyway, where are you going?
We had the room until 11. I'm going back to the office.
Oh, I see.
Just get on my desk.
So, can you hear me? Because you're frozen. Hello? Hello?
Hi.
Anyway, I'm really not, I don't know if you want
You really what?
to, I'm not sure that you really want to talk about Platt and Berman, because, I mean, the whole thesis is about modeling and measurement, and not so much on theories. So on your slide that said what today's hypothesis is,
Mm-hmm.
I didn't see really a sentence describing what the hypothesis is. I couldn't quite get it from your noun phrases.
Uh-huh.
You need to really clearly state the hypothesis, and at that point, if Platt and Berman have something to do with it, that's when you should say, here's where Platt and Berman fit in. But I don't see that.
Sure. I'll work on that. So
Major thing I would say about the talk is that, well, there's two main things. They're not that major, but I would talk about the differential encoding model and the Von stimuli before the developmental one.
why?
Because it seems like an easier story to tell to me. You started talking about measuring spatial frequency and using the hiddens as input to another network, but then you don't talk about that. Then you talk about the developmental model. I mean, I would go straight from describing the model to showing how it works.
Okay.
And then say, well, how can we get there? And then when you're talking about the developmental constraints, I would put side by side with the developmental constraints how you implemented them. So it wasn't clear what the connection with the developmental constraints was, and what that had to do with training something with low spatial frequencies.
Mm-hmm.
I mean, I know why it is, but if I didn't know, I don't think I'd know. So, I'm just saying to put a list, side by side. Here's the developmental constraint. Here's how I implement it.
Mm-hmm.
To start with the list on the left of the developmental constraints, and then click in to get how they're implemented, with maybe arrows if two things relate to one thing, like blurry being training earlier and low acuity.
Mm-hmm. What's the second major thing?
So I found it very weird to have the Ringo criticism after you'd already talked about the delay variability hypothesis.
Mm-hmm.
those seem to go together to me, right? I mean, and I wouldn't spend a lot of time talking about it. I would, just say that we showed this isn't it, and put a slash through it, and then say we think it has to do with delay variability, not just delay.
Mm-hmm.
I mean, then you can connect those two. I mean, you brought in the picture of the model in two very different points in the talk.
Right.
Didn't connect them at all.
I agree.
Um,
Okay. Anything else?
so I would put those back together.
Yeah, I mean, my concern was making the interlude too long, but yeah, if I try and shorten how much I say, I can keep it short and still not get too far away from the, asymmetry step.
Right.
Okay.
So I will send you my notes.
Mm-hmm.
I didn't tell you every little tiny note there was.
Sure. I appreciate that.
And then I would, I'd like to hear what the other people have to say, too.
We kind of disbanded, but I'll check with them.
Okay.
Because we got kicked out of the room. Thanks for the thoughts. Just one comment. I want to get your a little short discussion on. I think the Plout and Berman stuff is important to building a story and keeping things at the story level. And so I'd like to keep that stuff in. My goal will be to really clarify the relationship between the different ideas. You said you don't like theorizing. You just like and it wasn't so much in the dissertation. I mean, I think that's a weakness of the dissertation that I'd like to revise a bit, and I've done that a little bit. But I think to tell a story and to keep things at the story level and integrate the interhemispheric stuff with the asymmetry stuff, I feel much more comfortable with telling a story that involves the Plout and Berman stuff. Also because I think recognizing that there are these two different approaches in trying to pose what I've done is sitting in the middle. I think there's some value to that. So I'd like to try and go that route. What do you think about that?
I would like you to send me in the next hour a very clear story of how Plout and Berman relates to this.
Okay.
In 25 words or less. Maybe.
Wow. Sure.
If you can't state your idea in 25 words, you haven't got a clear idea.
Okay. And what I stated so far was not good enough.
Sounded great. I need to read it.
Okay. You got it.
And I'll take up to 50 actually.
That is very generous of you. Okay, so you send your things. I'm gonna ask around what other people thought, and then I will write this thing up for you and send it to you.
Yeah.
But I think it's pretty close. Much further along than yesterday.
Yeah, and I think it's good.
There you go. Thank you.
Especially, I told you yesterday I really thought it was really clear, showing how what you measured.
Right.
The question is going to come up at some point. How did you measure brain areas? Because you've got a ratio there of brain areas to 1, right? And
right. The great thing is I didn't have to measure, but yeah, I'm ready. I've got so many extra slides. Like the rest of my day is just going to be organizing them.
Well, don't forget to make some changes to those slides that you have already, right?
I was being facetious. Thank you.
Okay.
All right. Thank you.
Okay.
See you tomorrow.
So, Tamaki, do you want to just stay on and we'll have our meeting?
Yeah, that's fine, Join.
Okay.
Okay, bye. I haven't had a chance to read your NIPS submission yet, but I will. Can you hear me?
Yeah, well, I thought it was awesome, like compared to yesterday. It was, yeah, again, at least, it was much more organized, right? I mean, yeah, there were like couple things that, I wish that there were like more explanations. But yeah, I guess I'll talk to you a bit later.
Sure. Thanks. Yeah, that'll be helpful.
Uh, Hello?
Hi.
Hi. Okay.
So, what I was saying, listening, because you weren't plugged in yet.
Yeah, I
haven't
didn't have my headphones on. So.
Yeah, I haven't read your NIPS submission yet.
Okay.
But I will, and if you want, I mean, it might make better use of our time if I just did that now instead of having us meet. Unless you have.
No, not really. And yeah, I guess I'll also ask somebody like Vicente to read over it if he has time. So, because at least on the where I described a dictionary learning algorithm, I want that, yeah, I want to make sure all the notations and math is kind of right. So, yeah. But yeah, so yeah, at this point, as you can see, I don't have the results yet. As in, I haven't put the results in yet, but I think I'll try to kind of do the experiments again anyway, because I made some changes in the generated parameters and whatnot due to the flow on the way I've written, and I was also kind of trying to minimize the number of selection algorithm that I'll be introducing. So yeah, so there are some minor changes like that, but yeah, so once that's done, I'll put in the results. That will probably take another page and discussion where I'll talk about why some of the algorithms seem to work and also related works. So that should be like 8 pages.
And you get an extra page for your references now.
Right. So yeah, the references will be on the 9th page.
I noticed that they also said you can submit supporting material now. Which,
oh, is that the case? Huh.
Yeah.
Okay. Well,
they may or may not be reviewed. It may not be used in your review.
Yeah. Well, I suppose if I had some long proof or something, but yeah, I don't think I need one for now. But yeah, I'll see if I have like too many graphs or something. And yeah, maybe I'll just separate them. But yeah, okay. So please let me know if you have.
All right, so I'll spend the next hour or so looking at your paper, and then I'll give you some feedback on it.
Okay. Yeah. Yes. Please and let me know.
Well,
right.
Of course. Okay.
Thank you.
I'll see you later. Bye-bye.